We would certainly like to understand genes. We would like to understand them to the point where we know where they are, what chromosome there are, what their sequence is, so that we can then screen other individuals and see if anyone in the family has, say, a mutant version of this gene. We'd like to know what type of protein it codes for. Once we know that, we can do screening to find out where this protein is expressed and look at, say, mutant cases or individuals with disease to see if the protein levels have changed or if they're being expressed in new places. So this is what we would like to understand. One of the keys to all of this is finding the gene. All right, so if you were to say study fruit flies and you notice a mutant fruit fly, somewhere there is a mutant gene which is causing that abnormal phenotype. Where is it? If you don't know where it is, then you won't know what its sequence is. You won't know what protein it encodes. You won't be able to say, now that we know what its sequence is in the fruit fly, can we find a version of this in humans? If we know where it is in humans, can we screen a family to see if anyone's making abnormal versions of this protein? If we've just identified that it's a membrane receptor, could we search for pharmaceutical products which would interact with this membrane receptor that might cure this disease? You can't do any of that until you find the gene. So if you know where the gene is, there are all of these other things that you could then do. If you don't know where the gene is, you're extremely limited. And so then one of the things that geneticists do is they find genes, and then all of these other analyses then can start. How do you find genes? Well, there are lots of ways. And in this video, I'd like to talk about genetic mapping, where you would find a gene based on recombination and genetic experiments, as opposed to the next video where I talk about physical mapping. So one of Mendel's laws, the laws of independent assortment, is based on the idea that even though this individual has two dominant A alleles and two dominant B alleles in his cheek cells, his bone cells, etc., when he makes sex cells, each sex cell, each gamete, is haploid, having one of the copies of the A gene, but not both, having one of the copies of the B gene, but not both. If someone is a double heterozygote, in this case, a dihybrid, then when they make gametes, sometimes the dominant A allele is in the same gamete as the dominant B allele. Sometimes the dominant A allele is in the same gamete as the recessive B allele. Sometimes the dominant B allele is in with the dominant A, sometimes it's with the recessive A, etc. And so Mendel described this as independent assortment, so that the gametes weren't necessarily hanging out together. Now, Mendel described this before he knew what chromosomes were. No one knew what chromosomes were at that time. And so what we now understand is that, let's say the dominant A gene is here on this maternal copy of chromosome 1. The dominant B gene was on the maternal copy of chromosome 7. The recessive version of the A gene on paternal chromosome 1, the recessive one on maternal chromosome 1. Well, as you can see here, the pink and blue chromosomes that say an individual inherits from their mother or father don't necessarily have to hang out in a gang where they line up on the same side of the metaphase plate. If they line up on opposite sides, that means the future sex cells will then have the dominant A gene that someone inherits from their mother in the same gamete as the recessive B gene that one inherits from their father. The gametes which resulted from the other pairing would have the opposite. But that it doesn't have to work that way, where the two pink chromosomes, they could have lined up on the same side of the metaphase plate. So the sex cells which result could have both dominant alleles or both recessive alleles. So Mendel described this as independent assortment. And therefore, if two genes are independently assorting, then, for example, if you look at brown and scarlet in fruit flies, then in the F2 generation, you get a 9:3 to 3:1 ratio. This is the ratio indicative of independent assortment. So that it doesn't make a difference whether a gamete gets the dominant allele for gene A. The dominant allele for gene B doesn't necessarily follow it. Now, this is one of Mendel's laws, and so obviously this is worth discussing. However, it doesn't always work, and I know genetic students, if you have this understanding, I'm supposed to get a 9:3 to 3:1 ratio for independent assortment. I didn't get that ratio. Something must have gone wrong. There must have been a mistake. Well, very often, no. If you don't get what you expect, very often that means that something else is going on that you weren't originally studying, and that something else might actually be more interesting than what you thought you were studying. And so therefore, what would that mean if if we don't get a 9:3 to 3:1 ratio? Well, it could mean that the genes are linked on the same chromosome. So notice, if the dominant alleles are both on the pink chromosome, just one, and the recessive alleles are both on the blue chromosome, then they will tend to travel together because they are physically attached to each other. And so therefore you will not see independent assortment. They are not independent of each other, they are physically joined. And so the gametes which get the dominant alleles of for the red gene will get the dominant alleles for the yellow gene as well. And so if you're trying to find where a gene is, if you know where the red gene is, it's on chromosome 1. And if you can show then that the yellow gene is linked to it, you now know where the yellow gene is. It's on chromosome 1. You found the gene. Once again, that's a big deal. All right, if you can't find the gene, you're very limited in the genetic analyses that you can do. Once you find the gene, you have more possibilities. And so if these are stuck together, you will not get a 9:3 to 3:1 ratio. While they are stuck together, as was covered previously, in meiosis, homologous chromosomes do pair, and there is crossing over. So it is a process which must occur, that portions of the pink chromosome are exchanged with portions of the blue chromosome, so that while the red and the blue will tend to stay together because they are physically attached, crossing over, which occurs in the process of meiosis, can separate linkage groups. So groups, these genes don't have to be linked together permanently. And so that gets us to how we might do mapping in another species. Now, why do we care about fruit flies? Well, once again, if you can find a gene in a fruit fly, you can get its sequence, you now know what it looks like. You could then ask, do humans have a gene like this, and then find the human gene. So very often finding a gene in another species is then the precursor to finding the gene in humans. There are many of our own genes which we have found and we understand because they were studied in other species. So let's look, for example, at two genes, one causing a black body in Drosophila melanogaster, the gene is called black, and one causing vestigial wings. It is known as vestigial. So let's say that those are our genes A and B. If they were independently assorting, if they were not linked, in the F2 generation, you would get a 9:3 to 3:1 ratio of flies which were wild type for both alleles, which were mutant for the first gene black, but wild type for the wing gene vestigial, which were wild type for the body color gene black, but mutant for vestigial, and then black vestigial if you'd have both of these mutant conditions. So if you got a 9:3 to 3:1 ratio, you would know that these were not linked. They were either on separate chromosomes, or if they were on the same chromosome, they were extremely far apart, at which point they almost act as if they are independently assorted. But if they are next to each other, okay, if they're neighbors, then whatever gamete gets a dominant A gene will probably get the dominant B gene as well. And so as a result, you're not making all of these different kinds of gametes with different combinations of these alleles. The alleles stay stuck to each other. And then also then you're going to get fewer types in the offspring, because if you get a dominant allele for one gene, you will probably get the dominant allele for the second gene. And thus you'll tend to see a 3:1 ratio of flies which are either dominant for both or recessive for both. And so if you saw this in your F2 generation, you would realize, oh, these are linked, and they are so tightly linked that crossing over essentially never separates them. So if I know where one of the gene is, let's say I know where the gene black is, I now know exactly where the gene vestigial is, because they have to be right next to each other. Now, if you can find black flies with no vestigial wings, or vestigial flies with no black bodies, that means that crossing over has separated the two. So notice there's still a lot which are wild type for both. There are still a lot which are mutant for both. But when you start to get more of the intermediates, these can only exist if crossing over has separated the two alleles in gamete formation. And so if you're getting those other combinations, those are recombinants. The only way to get those is if a genetic recombination occurs. And it's just a simple principle now. The closer genes are, the less likely crossing over is to separate them. And so therefore, you could now count the number of these recombinants. The more recombinants that there are, the farther apart these linked genes are. You know they're on the same chromosome, but they're farther apart. When there are fewer of these flies, then that means the genes are closer together. Now, one of the things, I have a playlist on how to do a lab on this, doing what's called a 3-point cross. It's convenient to use genes which are on the X chromosome in flies. Now, this is a bit of an involved process, and I have an entire playlist which goes through this process. So rather than try to repeat everything there, I just want to hit the major point. And then obviously those who wanted to learn the procedure should probably watch the individual videos. But here we have a male fly, which is wild type for 3 traits. For eye color, it does not have vermilion eyes the way the female does. It does not have singed bristles the way the female does. That's the mutant case. And there are cross veins in the wings, but the female lacks those. So the female then is mutant for 3 traits, and the male is wild type for 3 traits. And then the question is, where are these genes? And so if you set up a cross where you have a fly with 3 mutations mating with a fly with just wild type for 3 traits, here you see their offspring. So here you see a fly which inherited the 3 mutant conditions from its mother. These are sex-linked traits on the X chromosome. And this female now has 2 X chromosomes, one inherited from the mother with 3 mutants, one inherited from the father with 3 wild type alleles. And in the body of the female, as she makes ova, then crossing over can occur. So while those genes used to be stuck together, all right, crossing over would separate them. And then what you could do is start counting flies. Most of the flies you have, if you're only counting the males, would either be all wild type for all three traits or all mutant. But what you would then observe is some of the flies would have some mixture of the wild type alleles and the recessive alleles. And if you count them, you could then come up with totals and realize that genes which are farther apart are more likely to separate through crossing over. The genes which are closer together are less likely to separate. And thus, by counting the number of flies, you can then get percentages, which then you convert to a map distance. And you could say, these two genes are so many map units apart, they're pretty far, because recombination separates them often. These two genes are close together, because crossing over rarely separates them. So as a result, you have a map of, say, the X chromosome. You can say there are so many, here, these two genes, vermilion and cross-veinless, they're the farthest apart. Singed is in the middle, but it's closer to cross-veinless than it is to vermilion. And I've done that by calculating the number of recombinants, because once again, the more frequently crossing over separates these to create recombinant flies, the farther apart they are. So now I've mapped the gene. You could then say sequence that part of the chromosome. And then once you get a sequence, you could say, all right, I know what this gene looks like. Do humans have any genes that look like this? And so mapping a gene in fruit flies could then lead to mapping a gene in humans. Now, obviously, sometimes we want to start with humans and try to map genes there, especially if you were, saying, here's a gene which influences depression or autism, or, something which is more human specific. We would not, be able to start with, say, flies, because obviously flies, would not have comparable phenotypes. And so how could we map in humans? Well, it's a similar principle in that in flies, you said here are these two mutant conditions, say having vermilion eyes and singed bristles. To what degree do they travel together or travel separately? Well, here we could do the same with a trait which we can measure. Let's say it's depression or a certain disease, where we could say here is the variant condition, here's the disease state, whatever. So we'll code that as yellow. And then here is the wild type typical condition, we'll code that as blue. Now, we're going to trace that through a family tree. And then we have to then compare that to another thing moving through the family tree, which I'm going to label pink. So this could be a certain sequence of DNA, or maybe it's an allele of a gene we already know, like blood type. We know, the allele for blood type is on this chromosome, and this person not only has this yellow trait, they also have blood type A. So we'll follow the yellow trait and blood type A going through the family, and we'll see to what degree they travel together. Now that works. Unfortunately, there's just not that many things like blood type or hair color where it's easy to trace. So very often this is compared to genetic sequences. So this person has an abnormal sequence at this spot on chromosome 1. So here's the question. If this is a sequence that we know and we can follow on chromosome 1, and here is a condition, a trait that we would like to map. Where is the gene that causes this trait? We can now look at the family tree and ask, do the people who inherit this pink piece of DNA, however we've identified it, where it's a part with a lesion or abnormal structure, or we can sequence it and they're getting this allele of this specific area. So some members of the family get the pink the pink piece of DNA. Some people in the family get the yellow trait. And then the question simply then becomes, are they the same people? Does the yellow trait travel with the pink piece of DNA? So does every person who gets the yellow trait, did they also then inherit the pink piece of DNA? In which case we would say, that they were linked and potentially tightly linked. But that's not the case here. So notice that while this person got both the pink piece of DNA and the yellow trait, this person got the pink piece of DNA without the yellow trait. This person got the yellow trait without the pink piece of DNA. So they don't seem to travel together. So if you're thinking that the gene that causes this disease is on chromosome 1, in this family, that's not the case, because the person who had both the disease condition and the pink piece of DNA, they did not travel together. So this disease is not linked to that spot. It is not on chromosome 1. Fine, so we do it again with a different chromosome. So let's imagine that this is a different chromosome then. And you could say, all right, is this gene linked to the pink piece of DNA, this part of, say, chromosome 6, which we can follow? Well, now notice what we're seeing is that this individual got both the yellow trait and the pink piece of DNA, yellow trait and pink piece of DNA. Same thing here, same thing here. So they do tend to travel together. Now, this person got the yellow trait without the pink DNA. This person got the pink DNA without the yellow trait. So they don't always travel together. They just usually do. So that means that they're linked. The reason that they're traveling together frequently is that they're all on the same chromosome, the gene that causes the yellow trait and this pink piece of DNA. But they're not at the same exact region. So the genes are linked, but they're not tightly linked. There's some distance between the two. That's why crossing over can separate them, and you can get individuals with the yellow trait without the pink piece of DNA, or the pink piece of DNA without the yellow trait. If, however, you had a family where you noticed that everyone who has the yellow trait is also getting the pink piece of DNA, not only does this mean that they're linked, they travel together, they must be physically stuck to each other, but they're tightly linked because we don't see any cases, or else maybe just very rarely do we see cases where you can be yellow without the pink DNA or a pink DNA without the yellow trait. So now not only are they linked, but they're tightly linked. So once again, the goal is to say, aha, I know where the gene is. It's on this spot of this chromosome. Once you know that, you can then do other tests. There's all of these, other opportunities which are open to you. And one of the ways of mapping a gene is genetic mapping. Unlinked genes assort independently, linked genes do not. And so then you're simply, whether it being in fruit flies or in humans using pedigree analysis, you're looking to see whether two genes are assorting independently or traveling together, or it may be a gene and then a specific piece of DNA, a sequence that you can screen for, whether they are traveling together. And this is a way of genetic mapping. And at the end of this, you could say, aha, I know where the gene is.